In this video, we will work through a sample MCAT passage. While MCAT passages often include questions that draw from multiple topics, this passage is focused on a single topic to demonstrate how the MCAT might present a specific concept within a passage or through its questions. Once you gain a stronger understanding of how topic focused questions appear on the MCAT, it is recommended that you practice with passages that test your critical thinking across a wider range of topics. To get the most out of this sample passage, be sure to read through the passage and answer the questions on your own before watching the full walkthrough. Let's get started and good luck. Positron emission tomography is a technique used for medical imaging. A PET scanner detects gamma photons that are produced when a positron annihilates with an electron. In this study, scientists used scandium-44, SC44, as a radioactive tracer attached to the Fab region of cetuximab. Cetuximab is a monoclonal antibody that binds to the epidermal growth factor receptor EGFR. It has been observed that many tumor cells express elevated levels of EGFR or its corresponding gene. The Fab region, or the antigen binding portion of cetuximab, was digested using the enzyme papain. The Fab fragment, along with a bifunctional chelator known as CHX-A-DTPA, was then bound to SC44. This three-component 5-tracer complex enables high-quality PET imaging of internal organs and tumors. SC44 is produced in a cyclotron by irradiating calcium-44 with protons through the nuclear reaction shown. SC44 has a half-life of approximately 3.9 hours, which is ideal for completing PET imaging procedures. It does not remain in the bloodstream for long. After injection of the three-component tracer, scientists were able to track its distribution across various body tissues, as shown in Figure 1. The term percent ID per gram represents the percentage of the injected dose per gram of tissue, a measure of how much tracer accumulates in specific region. Figure 1, tracer accumulation in body tissues. Question number 1. In this study, it is mentioned that the Fab tracer, scandium-44, SC44, is produced by irradiation of calcium-44 with a proton. Which of the following represents the correct nuclear reaction for this process? Question number 2. A patient is injected with 25 grams of scandium-44 bound to the Fab SHX-A-DTPA complex. Approximately how long will it take for the sample to decay to 5 grams? Question number 3. The Fab tracer includes a binding site that allows attachment to EGFR. Based on Figure 1, why does the percent ID per gram value remain elevated in kidney tissue compared to other tissues? Answer choice A says kidney cells express the highest level of EGFR. Answer choice B says kidney cells have a higher binding affinity for SC44 than other cell types. Answer choice C says kidney cells process the tracer to enable its clearance from the body. Answer choice D says kidney cells have a higher affinity for CHX-A-DTPA. Question number 4. In a follow-up experiment, scientists used different isotope of scandium Sc that undergoes alpha decay. They repeated the procedure to perform a PET scan, successfully binding this new scandium isotope to the Fab region of cetuximab and the SHX-A-DTPA chelator to form the same three-component tracer complex. However, after injecting this tracer into a patient, no binding of the Fab region to its target receptor was detected. Which of the following provides the most reasonable explanation for this observation? Answer choice A. Says the Fab region of the antibody was denatured prior to injection, preventing it from properly binding to EGFR. Answer choice B says the Fab fragment was not effectively digested by the enzyme papain, preventing the new SC isotope from binding correctly to the Fab region. Answer choice C says the new scandium isotope had a shorter half-life of approximately 3.1 hours, providing insufficient time to perform a PET scan. Answer choice D. The new scandium isotope underwent alpha decay, and the resulting decay products were undetectable by the PET imaging. Question number 5. Within liver tissue, what type of nuclear decay does scandium 44 undergo that allows it to be detected by a PET scanner? Answer choice A says beta plus decay. Answer choice B, gamma decay. C, alpha decay. D, beta minus decay. Here's the passage analysis. So on this slide, we will go over the passage analysis. On the MCAT, your notes should not be too extensive. Sometimes just writing one or two words is just enough. You only need to write enough to help you identify key information that brings you closer to the main idea of the passage. Every person takes notes differently, and which is completely fine. And on this slide, I'm going to present to you the kind of notes that I would take if I were to take the actual MCAT exam. What you should pay attention to is the structure of the passage. You should also note any science background information that you have learned before, recognize any relationships or comparisons, and of course, identify the main idea of the passage. So let's start. The first sentence reads positron emission tomography PET is a technique used for medical imaging. When you read this first sentence, a few things should come up to your mind. You should start forming a general idea of what the rest of the passage will discuss. Just from the phrase positron emission tomography, even without reading further, you can already predict that the possible direction of the passage would be. It might discuss radioactivity since it mentions positron. It may explain how a PET scanner works. It may describe how internal organs are analyzed, or it may outline some medical conditions that require PET scans. You do not need to write all these ideas, just to think about them mentally. It would help you focus your attention on what to expect next. The second sentence says a PET scanner detects gamma photons that are produced when a positron annihilates with an electron. This sentence is packed with background science information that you've likely encountered before. It mentions radioactive decay, where gamma photons and positrons, which is all the physics concepts that we have studied before. You should highlight this sentence and be ready in case a question later asks about them. The next paragraph says in this study, scientists used scandium 44, SC44, as a radioactive tracer attached to the Fab region of cetuximab. Cetuximab is a monoclonal antibody that binds to the epidermal growth factor receptor, EGFR. It has been observed that many tumor cells express elevated levels of EGFR or its corresponding gene. When you read this paragraph, you should recognize that the passage is shifting focus to the tracer used in the PET scan. Since scandium 44 is introduced here, make sure that you highlight it. This paragraph introduces new information, so note where they appear. The next paragraph says the Fab region or the antigen binding portion of cetuximab was digested using the enzyme papain. This Fab fragment, along with a bifunctional chelator known as the CHXA-DTPA, was then bound to SC44. This three-component Fab tracer complex enables high-quality PET imaging of internal organs and tumors. SC44 is produced in a cyclotron by irradiating calcium-44 with protons through the nuclear reaction shown. This next paragraph goes into more detail about how the tracer was prepared for PET imaging of internal organs. It contains a lot of detailed information, but the key point here is still scandium 44 because it is repeatedly mentioned, so you can predict at least one or two questions will be about this isotope. The paragraph also hints at a nuclear reaction, which is not in a form that is very familiar, so be alert about a question that, that will test your understanding of it. The final paragraph says scandium 44 has a half-life of approximately 3.9 hours, which is ideal for PET imaging procedures. It does not remain in the bloodstream for long. After injection of the three-component tracer, scientists were able to track its distribution across various body tissues, as shown in Figure 1. The term percent ID per gram represents the percentage of the injected dose per gram of tissue, and which is a measure of how much tracer accumulates in a specific region. The final paragraph again provides more information about scandium 44, confirming that this isotope is central to the passage. Since we just studied half-life, take note that this concept may appear in one of or two questions. So, on your first read, you don't need to thoroughly analyze the figure. Just read the caption to understand what the figure is showing. If a question asks about the figure, you can return to it and then analyze it in more detail at that point. The main idea of the passage is that it describes the radioactive tracer used in positron emission tomography and how it functions in imaging studies. Let's move on to question number 1. Question number 1 asks, in this study, it is mentioned that the Fab tracer scandium 44 is produced by radiation of calcium 44 with a proton. Which of the following represents the correct nuclear reaction for this process? The passage mentioned scandium 44 multiple times, and we predicted that one of the questions would ask about this isotope. And the first question asks us to write a balanced nuclear reaction that produces scandium 44. The passage provides a reaction for making scandium 44, but it is written in a unfamiliar form. Let's use what we know about nuclear reactions to pick out the correct answer. From our prior knowledge, we know that every nuclear reaction must conserve both atomic number and mass number on both sides of the equation, or both sides of the reaction. We can use this rule to eliminate wrong answer choices. Starting with option A, both atomic numbers and mass numbers are balanced. However, the option produces scandium 45 instead of scandium 44, which was stated in the question stem. So option A is not right. In option B, the total mass number on the reactant side is 44 plus 1, which equals 45. But scandium 44 has a mass number of 44 on the product side. Because the mass numbers do not match, option B is wrong. Option C incorrectly represents a proton. A proton has a mass number of 1 and an atomic number of 1. But this option shows the wrong values, so option C is not right. The only option that conserves both the atomic number and the mass number is option D. This reaction includes a neutron as a product, which balances the mass numbers perfectly. While we may have never seen a reaction that emits a neutron, this is completely valid. As long as we apply what we have learned before, we can confidently identify the right answer choice. So the correct answer is D. As you can see, this question was not difficult at all. It may have been presented in a slightly obscure way, but if you stay grounded in the basic concepts and trust what you have already learned, you will find the correct answer choice becomes clear almost every time. So, with that mindset, let's move on to question number 2. Question number two says a patient is injected with 25 grams of scandium 44 bound to the Fab SHXA, the chelator complex. Approximately how long will it take for the sample to decay to 5 grams? This is a very straightforward problem that asks us to solve a half-life problem. The question provides the initial amount of the sample, and we are asked to determine how long it takes for that sample to decay to a final amount of 5 grams. The passage states that the half-life of the sample of scandium 44 is 3.9 hours. We will use the conceptual method, which uses the arrow diagram to solve this problem. So we need to start with the 25 grams of scandium 44. After the first half-life, the amount is reduced to 12.5 grams. After the second half-life, it decreases further to 6.25 grams. And after the third half-life, it becomes 3.125 grams. Since the final amount given in the question stem is 5 grams, this value lies between the second and the third half-lives, and it is much closer to the second half-life. On the MCAT, we typically don't need to calculate exact values, and approximate or ballpark values is just sufficient, especially since the answer choices here use the word about. So to estimate the total time elapsed, we multiply the number of half-lives by the length of one half, or 3.9 hours. Because we do not have a calculator on the MCAT, we can round 3.9 to 4 hours for easier estimation. So for 2 half-lives, 2 times 4 is equal to 8. And for 3 half-lives, 3 times 4 is equal to 12. Since 5 grams lies between 6.25 grams and 3.125 grams, the total time elapsed should be between 8 and 12 hours. It should be a little bit closer to 8 hours. Therefore, the best answer choice is option B, or about 9 hours. So here's question number 3. It reads the Fab tracer includes a binding site that allows attachment to EGFR. Based on figure 1, why does the percent ID per gram value remain elevated in kidney tissues compared to other tissues? So this question refers to figure 1, which shows how much of the tracer measured in percent ID per gram, or percentage of the injected dose per gram of tissue, is detected in different body tissues over time. So even though the tracer binds to EGFR, the passage does not mention how much EGFR each tissue expresses. So let's quickly go through the options. In option A, it is stated that the kidney cells express the highest level of EGFR. This is wrong because the passage explicitly states that it is the tumor cells that express the highest levels of EGFR. B says kidney cells bind more SC44. This option is also wrong because SC44 does not bind EGFR. So option C is correct because kidneys process the tracer to clear it from the body. Kidneys filter blood, removing waste, so it makes sense that the tracer levels would stay higher in the kidneys. Option D states kidneys would have a higher affinity for the chelator, the CHXA DTPA. This statement is not mentioned in the passage. When you see a question like this, so don't overthink the details and just stay focused on what the passage tells you. Apply what you know about basic science and use the process of elimination to find the right answer. So the right answer to this question is answer choice C. Let's move on to question number 4. Question number 4 was a bit long, and it says in a follow up experiment, scientists used different isotopes of scandium that undergoes alpha decay. They repeated the procedure to perform a PET scan, successfully binding this new scandium isotope to the Fab region of cetuximab and the CHXA DTPA chelator to form the same three component tracer complex. However, after injecting this tracer into a patient, no binding of the Fab region to its target receptor was detected. Which of the following provides the most reasonable explanation for this observation? Remember from the second sentence of the passage that a PET scanner detects gamma photons released when a positron annihilates with an electron? That means PET detection relies on positron emission, not alpha decay. So in this case, the new isotope of scandium undergoes alpha decay. So it produces a helium-4 nucleus. It does not produce a positron. So since no positron is released, no annihilation takes place with an electron and no gamma photons are produced. So the PET scanner cannot detect the tracer. This matches answer choice D. So the answer choice D is the correct answer to this question. This question shows why it is important to analyze the passage as we did. Remember when we said that the second sentence of the passage could be important? Here's where that pays off. Always note science facts, relationships, or comparisons. And also note for any repeated ideas. They often show up later in questions. So let's move on to the final question of this video, number 5. Number 5 says within liver tissue, what type of nuclear decay does scandium 44 undergo that allow it to be detected by a PET scanner? This question is actually asking how does scandium 44 produce a signal that the PET scanner can detect? The passage tells us that the PET scanner detects gamma photons from the annihilation of a positron with an electron. The name positron emission tomography also hints at that same answer. So scandium 44 undergoes beta positive or beta plus decay, producing a positron that annihilates with an electron and it emits a gamma photon, which is detectable by a PET scanner. So the correct answer to this question is the answer choice A. I hope you were able to learn something new from this video. Remember, success on the MCAT requires a strong understanding of the MCAT exam's language, which means being well versed in the underlying science concepts. Having a solid conceptual foundation is essential for effectively approaching the critical thinking questions the MCAT presents. If you'd like to review the concepts covered in this video or work through additional practice problems, be sure to check out the other videos in this playlist or the links in the description below. Thank you for watching and keep practicing. Every practice session brings you one step closer to your target MCAT score. Good luck on your MCAT journey.